lifestyle of some male homosexuals has triggered an epidemic.
A third of people who need life-saving HIV drugs are getting treated worldwide.
There are an estimated 91,000 people living in the UK with HIV. This includes a figure of 22,000 people who are estimated to be infected but are undiagnosed.
A few months ago, in a biotechnology research centre in Spain, the first major steps were taken towards producing a viable vaccine to eradicate one of the most prominent diseases of the 21st century. So how does it work?
The MVA-B HIV vaccine is an altered or attenuated smallpox virus which cannot replicate in our cells. The vaccine particles contain genes inserted from HIV, causing it to display HIV-specific proteins on its surface. This provokes an immune response. Dendritic cells engulf the particles, break them up, and display the HIV-specific protein molecules on their surfaces. Helper T cells with receptors specific to the proteins bind to them. However, finding T cells in the body with receptors that fit these HIV proteins can take a few days. When the helper T cells have bound, they release cytokines, which activate other white blood cells. T white blood cells differentiate into T killer and memory cells. Helper T cells also activate B cells, which form memory B cells and plasma cells, able to produce antibodies specific to the displayed HIV proteins. The plasma and T killer cells produced remove the vaccine particles from the body and are short-lived, but memory cells, able to recognize HIV proteins, may remain in the body for years. This means that in a person infected with an HIV virus displaying the same proteins as the vaccine, the immune response will be much faster, and the individual will be more likely to beat the infection.
HIV infects T white blood cells, incorporating its DNA into theirs. Developing an effective HIV vaccine is extremely difficult because the virus attacks and hides within the very immune cells that would otherwise destroy it. HIV occurs in many forms and is constantly changing, which any treatment needs to be able to keep up with. Although the MVA-B vaccine is only effective against the European HIV subtype B, Dr. Esteban tells us that his laboratory have also developed the vaccine MVA-C from subtype C prevalent in Asia and Africa. But these treatments are not going to be available anytime soon. Dr. Esteban comments that research towards the vaccine that reduces infection by at least 50% in a much larger group of people, being optimistic, will take between 5 and 10 years. Currently, the vaccine also requires repeated booster immunisations to maintain protection. MVA-B is obviously not yet perfect, but it is a step in the right direction.
Testing of the MVA-B vaccine was initially undertaken against simian immunodeficiency virus in macaque monkeys and was found to be 100% effective in creating an immune response to SIV infection, although this has been the case in other vaccine trials subsequently found to be ineffective against HIV. Phase 1 testing in humans was then able to proceed.
The testing was carried out on 30 HIV-free individuals as the vaccine was still part of Phase 1 trialling. The principle behind the vaccine was to train the immune system into remembering the virus. 24 of the volunteers received the vaccine, 6 received a placebo. The volunteers were inoculated, and over the following months, blood samples were taken and tested to see whether there was an immunological reaction. The researchers found increasing levels of B lymphocyte, proving that the vaccine was boosting the immune system's response. 90% of the volunteers showed an increased immunological reaction to the HIV virus. 85% of these kept up this reaction for a year.
Future testing needs to ascertain whether a therapeutic effect is seen in HIV sufferers, reducing viral count and progression to AIDS. In this instance, HIV cannot be removed. The viral genes would remain within a cell's DNA, but would be controlled by the vaccine. Only if MVA-B succeeds in these initial therapeutic trials can it go on for more rigorous testing as a preventative HIV vaccine.
Current treatment for HIV consists of therapy with antiretrovirals, a collection of drugs which disrupt replication of the HIV virus. However, resistance to antiretrovirals can develop, so drugs may be administered in combination to decrease the chance of this occurring, a method called highly active antiretroviral therapy. Antiretrovirals can have very unpleasant side effects and additionally cannot remove HIV from the body. An effective HIV vaccine would therefore be a major breakthrough and is seen by some as the only real way that the AIDS pandemic can ever be stopped. The MVA-B vaccine is not the only potential HIV vaccine being tested. In fact, since 1987, over 30 prototype vaccines have been produced. Such vaccines may be either preventative, stopping infection from occurring, or therapeutic, which lessen the effect and infectivity of HIV. At present, the only effective preventative methods involve barriers which prevent transmission of bodily fluids from one person to another, and in the case of HIV, prevention is very obviously better than cure.